So hello everyone and Happy New Year or gelukkig nieuwjaar for all the Dutch people in the room. Happy to see many familiar faces, but also new names there in the virtual room. And thank you for joining. My name is Juana Georgiana Roos-Oswald. I am a postdoctoral researcher in the BioClock Consortium. And together with Hannah Brooks, which is a PhD student in the same consortium, we will host the session today. So welcome back to this new series of the lectures. In this new series, we dare to also invite speakers outside of our bubble, outside of the BioClock Consortium bubble. Another new thing this year is that our speakers also bring a bit of their professional career story in their talk, which we think might be beneficial and inspiring, especially for all early career researchers. So thank you also to our first speaker this year to dare to do that. Today, our speaker actually sacrifices her evening, actually sacrifices her early morning to talk in our BioClock Academy lecture, and we thank you for that. So it's challenging maybe for your chronotype, you can tell us later, but I don't want to spoil it. And just give you some more organizatoric points for now. So the BioClock Consortium is a Dutch research organization which was set up to study different aspects of the biological clock in human, in health, and in society. And the BioClock Academy is a lecture series organized within this consortium. And it is addressed to everyone who is interested in chronobiology, but also very specialized on early career researchers, because we want to learn. So there's a lot of PhD students hearing these talks. So its aim is to introduce you to the basic concepts and equip you with the proper theoretical knowledge by experts in the field. The lecture series takes place every third Wednesday of the month. And for the time frame for today, we will do the following. So we will have a 40 minutes talk, followed by 15 minutes of discussion. So roughly 1 hour, so that we can close the session at 5 p.m. our time. During the talk, you can write your questions in the chat. We will keep an eye on that. But you can also afterwards unmute yourself at the end and ask the speaker. So now I want to give the floor to Hannah, which will introduce the speaker of today.
Yes, hello everyone. So also a Happy New Year from me. And I'm very pleased to introduce our first of the year speaker, Dr. Renske. She will be guiding us through all the effects that light can have on alertness and thermoregulation, a topic on which she already started working during her PhD at the University of Groningen. And currently she's working as a postdoctoral fellow at the Department of Psychiatry and Behavioral Sciences at Stanford University. And here her work is mainly focused on investigating the association between sleep patterns and health issues, and also on the influence that light can have on human sleep. And based on the rewards she got for her previous work and also her brief contact before, I think we have a very enthusiastic and engaged chronobiologist in front of us. So I think we will have an inspiring talk. So Renske, please go ahead.
Hi, good morning, everybody. It's 7 a.m. here. And while I'm not an evening chronotype, I'm definitely more of a morning chronotype. I'm still a tiny bit sleepy, so I'm going to try and do my best. I'm very excited to be here, though, and thank you for having me. I'm going to talk a little bit about the circadian nexus, investigating the impact of light on alertness and thermoregulation. And because I couldn't resist, I also snuck in a little bit of the light effects on sleep, because I think it's such a fascinating topic. Feel free to post questions in the Q&A, and I guess we will go through them at the end. So as maybe all of us know, I know everybody here kind of works on different organisms. Humans are diurnal, and what that means is that humans have a consolidated period of rest, usually at night, and a consolidated period of activity, usually during the day. So that very much coincides with the natural light-dark cycle of when the sun is out and when the sun goes down. But our relationship to light very much changed once electrical light emerged, right? So many years before Christ, there was fire. At some point, there was gas, and then incandescent lighting started to arise, fluorescent lighting and LED lighting. And basically what this means is that we're no longer subjected to what the light-dark cycle tells us to do. So basically, when the sun sets, we can extend our activity because we have electrical lighting. And this is great for many aspects of life and quality of life, but we're starting to see more and more that this is also detrimental for certain aspects of life. And it's really interesting because if you look at the first advertisement of electrical room of the Edison bulb, it actually says the use of electricity for lighting is in no way harmful to health, nor does it affect the soundness of sleep. And this is something that we're going to explore a little bit more during this talk, because I think it's a bit more nuanced than how it was postulated back then. So for those who do not know, as it's such a mixture of people in the audience, I'm really going to talk about the non-image forming effects of light. And what that means, it's like effects of light that go beyond vision. Because of course we know that light enables us to see, light enables us to do things, but light also has effects that extend beyond vision. And if we look at the eye, so here's an image of the eye where light enters the eye through the lens. And there at the back of the eye, there is the macula, and there's a bunch of cells in there, including IPRGCs, rods, and cones. And all these cells have different spectral sensitivities. And what I mean by spectral sensitivities is that light comprises of different wavelengths. And these rods, cones, and IPRGCs have a different sensitivity to different colors of light. So there are type of cones, the short cones that are sensitive to light of shorter wavelength. The rods are basically intermediate wavelengths. You have the medium type cones that are sensitive to light around 550 nanometers, so intermediate wavelengths, and the longer cones, so light that is red, longer wavelength. And then the IPRGCs is the special type of subset of cells that is mostly sensitive to melanopsin. So that peaks around 480 nanometers. So basically light and light as what I am in, white light, comprises of a little bit of all these colors and stimulates all these cells. And why this is so important is because the rods and the cones are predominantly implicated in visual responses to light. So they really enable us to see, and more research highlights that actually IPRGCs are also important for vision. But one of the main functions that IPRGCs have is to contribute to the non-image forming effects of light. And basically it means that when light enters the eye, it goes through the retinal hypothalamic tract, which is an amazing word for Scrabble if you ever want to play. And through the retinal hypothalamic tract, it reaches the suprachiasmatic nucleus. And for most, this must be like a very familiar nucleus because it's the main biological clock for humans, at least. So these IPRGCs, this specific subset, is very important for this trajectory through the suprachiasmatic nucleus. And it's really interesting because we're starting to see more and more and discover more and more about these IPRGCs. And this is not my own work, but this is actually beautiful animal work coming out of Schmidt's lab. And basically it's showing that there are many subsets of IPRGCs, of these specific cells for non-image forming information, and that they project to many different nuclei in the brain. And that includes the SCN, so the suprachiasmatic nucleus, but also lots of other nuclei. For example, the VLPO, which is implicated in sleep, the lateral hypothalamus, which is implicated in alertness, and so on. Basically meaning that it's very likely that light has an effect on non-image forming responses because it's connected and it's projecting to so many nuclei in the brain that are implicated in this. So today, as I said, I'm going to talk a little bit about the effects of light on human alertness, a little bit about sleep, and a little bit about thermoregulation. So let's start with alertness. So what is really alertness? It is quite hard to define alertness, actually, because it's really a feeling, right? Like I feel alert at this time of day. But clearly, it's really defined as a state of being vigilant, watchful, and attentive to one's surrounding. It's defined as mentally aware, and you're ready to respond quickly to stimuli, potential dangers, or changes in the environment. And an alert person is typically focused, observant, and able to react promptly to various situations. Alertness is essential for maintaining awareness, for making decisions, and ensuring a quick response when necessary. So basically, if a puma were to walk into my office right now, I can run away because I'm very alert. And how in the lab we usually measure alertness is in a multi-measure approach. And that means that we try to capture the concept of alertness through relying on many measures, including self-reports. So we will ask you, hey, how alert do you feel on a validated scale, obviously? We try to assess how alert you are through objective measures. So how well do you perform on a cognitive task? And we measure your physiology. So usually we rely on brain waves. So what does electroencephalography, so placing electrodes on your skull and picking up on these brain waves, what do they tell us? And usually we try to combine all these aspects to capture the concept of alertness. Now, to talk about the alerting effects of light, we are definitely not the first. So there is this beautiful work from the early 2000s in which they designed a dose-dependent relationship between light intensity and alertness. So what does that mean? So here on the x-axis, we have light intensity on a logarithmic scale. And on the y-axis, we have subjective rated alertness. And a dose-dependent relationship means that at low light intensities, there's no effect of lighting on alertness. So under low intensity lighting, researchers ask the participants, how alert do you feel? And the participants say they feel okay. They don't feel so alert. When they increase the light intensities, there's a steep increase in how alert people start to feel. So if you go from a dimly lit room to a very highly lit room, people become a lot more alert. And at some point, this relationship kind of saturates. So at some point, it means that if you increase the light intensity even further, it's not going to make a difference for how alert you feel because you already reached the peak of your alertness. And this is what we call a dose-response relationship. Low intensity, no effect. High intensity, there's a big effect, and it saturates out. And this is what happens in subjective alertness. So how alert do you rate yourself? But also in objective correlates of alertness, such as EEG. And this is an opposite relationship. So a higher alertness is lower on the scale. And basically, we see the same trajectory here. So there's a really nice dose-dependent relationship between light intensity and alertness at night. But humans are diurnal and we are active during the day, right? So it's much more important and practical to know if such a dose-dependent relationship also exists during the day. Because in the end, this is when we are active. This is when we are presenting and doing our job. So we need to be alert during the day when we're up and awake. So if we try to replicate this beautiful dose-response curve at night during the day, it's really, really hard. And we try to do this at many times of day. So we try to do this in the early morning, in the mid-morning, in the early afternoon, and in the late afternoon. And we tried this in a large sample size under semi-controlled conditions. So we controlled how much they slept, the things they were doing. And still we cannot replicate this dose-dependent relationship. So we use the same light intensities at the study at night, actually a little bit higher. And we could not find a dose-dependent relationship between subjective alertness and light intensity. Basically meaning that we could not replicate the dose-dependent relationship at night during the day, suggesting that light during the day cannot really make you more alert. And to be really, really thorough, it wasn't just light intensity, but also the spectral composition. So in a different study, we exposed participants to lighting that was just fluorescent, so the type of lighting that I am in now, normal white light or blue enriched white light. And if you remember, this all has to do with the sensitivity of these rods, cones, and IPRGCs in our brain, knowing that IPRGCs are mostly sensitive to light of bluer spectral composition, so the blue enriched white light. And we found no significant differences. So it doesn't matter if you're in light that is just white, that is a little bit bluer, or that is very, very blue. You cannot induce alertness during the daytime in somebody who is well rested. So then it becomes kind of an issue, right? Because you have this beautiful dose-dependent relationship between light intensity and alertness at night, but it does not replicate during the day. So why? What is it about alertness that is different during the day than it is during the night? And this all has to do with the circadian component in alertness and the homeostatic component in alertness. So this is the good old two-process model of sleep designed in the 80s, which states that most components, so most concepts, including alertness, have a homeostatic component and a circadian component. And the homeostatic component is depicted in the thick lines. And it basically means from the moment that you wake up, you have an accumulation or a buildup of homeostatic sleep pressure. And as long as you stay awake, this sleep pressure starts to build up more and more and more. So there's an accumulation here. And when you go to sleep, the sleep pressure dissipates. So this is process S, or homeostatic sleep pressure. And in addition to that, we have, of course, the process C, the circadian clock, which cycles with a duration of approximately 24 hours. And it means that at certain times, the circadian clock will be promoting alertness, and at certain times, the circadian clock will be promoting sleep, because that's what our internal clock tells us to do. And these two concepts are highly, highly intertwined. So, I mean, I just woke up, so my homeostatic sleep pressure is relatively low, and my circadian clock is promoting wakefulness. But in 16 hours, my homeostatic sleep pressure is going to be super high because I've been awake for the whole day, and my circadian clock starts to promote sleep. And this is the time where I will be able to fall asleep. And if we compare the studies, so the dose-dependent relationship at night, which was measured here when homeostatic sleep pressure is really high and the circadian clock is promoting sleep, compared to our studies that we conducted, where we measured during the daytime, homeostatic sleep pressure is relatively low and the circadian clock is promoting alertness. So if you want to figure out what is the difference between, the effects of light at night versus during the day, we have to balance these two concepts, because is it homeostatic sleep pressure that's different, and it's the circadian clock that's different. So if you want to disentangle these two concepts, you have to conduct something that is called a forced asynchrony experiment. And I will spare you the details because forced asynchrony experiments are highly, highly complicated. But the bottom line is you have participants that you keep in your human isolation facility for 5 consecutive days. So they are in our human isolation facility for 5 days under an alternative light-dark cycle. So they sleep for 5 hours and they're awake for 13 hours. And this is a double plot, which is common in circadian sciences. Basically, all you have to know, participants stay up for 13 hours, then they sleep for 5, they stay up for 13 hours, and they sleep for 5. And this means that light exposure, because during this time they're exposed to lighting, this means that sometimes light exposure occurs during the circadian day, so which would be, the daytime if they were under normal light-dark cycle, and sometimes during the circadian night. So sometimes when the circadian clock is actually promoting sleep, they will be exposed to lighting. And we conducted this experiment in 8 participants, and they once did these 5 consecutive days under dim light conditions of like very low intensity lighting, and once under high intensity lighting. So we had some really nice participants to be locked up in our human isolation facility twice. So then we can mathematically disentangle the effects of how long you've been awake from the effects of the circadian clock. And I will show you a little bit of the data. So if we look at the effects of subjective alertness, and we'll start here. So this is the effect of process S, homeostatic sleep pressure. And we can see that when participants wake up, they wake up relatively alert. And when they go to bed, 13 hours later, we can see that they're relatively sleepy. So this is their subjective rating, right? With higher alertness, lower on the scale. So they wake up alert and they go to bed sleepy, which makes sense because they've been awake for 13 hours. If we look at cognitive performance, on the other hand, and these are 2 simple reaction time tasks, a PVT and a go, no go, it's basically simple assessments of cognition, we can see a completely different pattern. So participants perform a certain way when they wake up, and they perform approximately the same way when they go to bed. And this means that there's not really an effect of how long you've been awake on simple reaction time tasks. So this is really interesting, because if we try to capture alertness in a multi-measure approach, so through subjective alertness ratings and performance, cognitive performance, they do not point in the same direction. So we have to take these two apart. Now, if we throw light in the mix, what do we get? We can see that people under high intensity lighting, they also wake up quite alert and they also go to bed quite sleepy. But the trajectory from alert to sleepy is a little bit different. So people under high intensity lighting, they stay alert for longer. So they feel alert for a longer amount of time before they also get sleepy when they go to bed. And then if we look at cognitive performance, we can see that they actually perform a lot better always under high intensity lighting. So always under high intensity lighting, they have better reaction time tasks, suggesting that they are feeling more alert and that they're cognitively performing better. So it's a significant difference between their subjective ratings and how they perform. Now, if we look at the effect of the circadian clock, what do we get? So there's a beautiful circadian mechanism, and on the x-axis here we see circadian clock phase, in which circadian phase 0 means dim light melatonin onset, so the timing of the internal clock when we start to produce melatonin. And we see that there's a significant decrease in alertness after melatonin production and an increase in alertness later on. And this circadian effect is less pronounced in cognitive performance. And now if we throw in light exposure, again, people seem to stay a bit more alert under high intensity lighting. So the circadian effect is a little bit less pronounced. And this is actually the same in cognitive performance. So people perform a lot better dependent on circadian clock phase under high intensity lighting. So now if we combine these results, so if we throw it all back together again, here on the x-axis, we have circadian clock phase, so the timing according to your own internal circadian clock. And on the y-axis, we have how long you have been awake. And then these green and red squares, they indicate if there is a light-induced improvement or a light-induced decrement. So green means a light-induced improvement, and red means a light-induced decrement. And we can see that there's a couple of green squares for subjective alertness, which means that yes, light can induce subjective alertness, but only at very limited times of day. And here in red, we see the projected daily time course, meaning that, hey, if we put our results back together, and if you wake up at 7 a.m. and stay awake for the next 13 hours, this is what your trajectory would look like. And we can see that indeed, there's no, like the red line doesn't hit any of the green squares, meaning that, yeah, light, high intensity lighting during the day cannot induce subjective alertness. However, if we look at cognitive performance, the red line hits tons of green squares, meaning that even though you don't feel more alert, you actually do perform better if you're exposed to high intensity lighting during the daytime. This is a really interesting finding. And actually, we were able to replicate this depending on the spectral composition of lighting. So I showed a little bit of this data earlier. So again, this depends on, the activation of these IPRGCs. And basically, it means that if you're exposed to light of bluer spectral composition, you will perform better also on complicated cognitive performance tasks. So these are tasks that measure visual scanning, working memory, or recognition of facial emotions. And we can see that you do better on these tasks if you're exposed to light of bluer spectral composition, so light that activates these IPRGCs. And again, this does not change your feelings of alertness, but you will perform better. So actually, how I was saying that there's a multi-measure approach in assessing alertness, it's not really true, because cognition and alertness clearly can do very different things. And if we want to assess alertness, we have to take these concepts apart. So then I talked a little bit about sleep and sleep duration and how this affects your alertness. So what about the light effects on sleep? And why I think this is such an interesting topic to talk about is because you almost cannot open the news, at least here in the US, or there's a new article about how light at night influences your nighttime sleep and how it's bad for you. And of course, I cannot argue with that logic, but one thing I do want to indicate is we're really battling human behavior, right? It's not just the effect of light being there and this affects our sleep, but it's really light extending our activity window and enabling us to stay up after dusk. And we're battling with human behavior because companies like Netflix, they tweet things like, oh, sleep is my greatest enemy. And they're absolutely right, because who doesn't compromise a night of sleep for binge watching a show? But there are other effects of light on sleep. So first, let's define sleep. So sleep, it's a natural and recurring state of reduced consciousness and inactivity. So when you're asleep, you're reduced. You're not as alert as you are when you're awake. It's characterized by decreased sensory awareness and a temporary suspension of voluntary muscle activity. And it's essential for overall health and well-being. And we're starting to explore more and more of these beneficial effects of sleep. It plays a crucial role in processes such as memory consolidation, immune system functioning, and physical restoration. In other words, sleep is very, very, very important. Sleep consists of different stages, including rapid eye movement sleep and non-rapid eye movement sleep. Each serves a specific role. And if we measure sleep in the lab, we also usually use a multi-measure approach. So we do polysomnography, we attach electrodes to the skull, and we pick up brainwave activity and self-report. So how do you feel you slept? And if we go back to our within subject force synchrony, in which we expose participants to lighting at different circadian clock phases, we also measured how they slept after that. And we can see how does light influence sleep. And here I picked apart these brain waves in the three different stages. So here we have the non-REM sleep, which we for simplicity reasons will call deeper sleep, the REM sleep, which is lighter sleep, and wakefulness. So how much did you wake up after you went to bed? And if light exposure occurred when the circadian clock was promoting wakefulness, there's not really a difference between the amount of deep sleep, lighter sleep, or wakefulness after bright light exposure or dim light exposure. But if light exposure occurs when the circadian clock was actually promoting sleep, there's a significant difference. So people who were exposed to a lot of light when the circadian clock was actually promoting sleep, when they do go to sleep, they sleep deeper. So there's more deep sleep and there's less wakefulness, meaning they sleep more consolidated and they sleep more deeply. And this also occurs when the circadian clock is partially promoting sleep and partially promoting wakefulness. And then if we again look at when the circadian clock is promoting wakefulness and light exposure occurs, this effect disappears. Meaning that if you're exposed, so if you do work at night and you're exposed to a lot of light, your sleep afterwards will be more consolidated and more deep. And this is not just what we pick up in the brain, so brain waves, but it also, if we ask you how you feel you sleep. So this is the Groningen sleep quality scale. And this is a little bit hard to interpret, but all you really need to know is that better sleep quality occurs at higher ratings. And basically, you see this beautiful parabolic relationship, meaning that if you're exposed to lighting when the circadian clock is actually promoting sleep and you go to sleep after that, you actually rate your subjective sleep quality better. So you feel like you sleep better if you're exposed to a lot of light when you're awake at night. And this coincides beautifully with what we pick up in the EEG. So this is one of the examples where our multi-measure approach works out, because the EEG tells us you slept better and you rate like you sleep better. You feel like you slept better. And of course, a prime example of people who go through this on a regular basis is people who do shift work. So one argument for being exposed to lots of light when you are on your night shift is actually that you will sleep better after that, and you will feel like you slept better. And we all know is that if you sleep better, you will feel better the next day. So this is a really interesting finding of how light can influence sleep. So what about thermoregulation, and how do all these concepts tie in together? So what is thermoregulation? It's the physiological process by which an organism regulates its body temperature to maintain an optimal internal environment. And thermoregulation is absolutely crucial for properly functioning of bodily processes. I mean, it's really important for the functioning of enzymes, and they often have very specific temperature ranges at which they work. So human thermoregulation and actually animal thermoregulation is extremely important. In warm environments, thermoregulation helps dissipate excess heat to prevent overheating, usually through mechanisms like sweating and vasodilatation, expansion of blood vessels, and getting rid of heat. In cold environments, it involves conserving heat through processes such as shivering and vasoconstriction. So your blood vessels constrict, so you keep all the heat inside. In the lab, we usually measure thermoregulation in two ways. So we try to measure core temperature, so what goes on the inside, and usually through an ingestible pill, so a temperature sensor, or through rectal temperature, so you can use a rectal probe. We also try to measure skin temperature by putting little measurement buttons on the skin at various places to pick up if there's vasodilatation, so heat dissipation, or vasoconstriction, so keeping heat in. And just a small reiteration. So core temperature is really heat production and heat dissipation because of internal organ activity, whereas what we measure on the skin is usually heat dissipation and heat constriction. So our eye buttons, so the measurements on the skin temperature, we usually place them on the hands and the feet, which is great regions for heat dissipation and heat constriction. And here's a small indicator of how this really works. So if you're really, really warm, we have these blood vessels that work as a shunting mechanism. And if you're warm, this shunt goes open, meaning that blood can circulate through all the small vessels, mostly in the hands, the palms, and in the feet. And this is how we reduce heat. And if we're really, really cold, these blood vessels will constrict, and it means that we are able to keep heat in, so heat conservation. So what about thermoregulation, alertness, and core body temperature? Well, they actually are all very much interrelated. So if you remember, I showed this image of the circadian component in alertness with a beautiful peak after melatonin onset and better alertness after that. Actually, core temperature shows a similar circadian component, but opposite. So your core temperature decreases after melatonin production and increases afterwards. So it's the opposite process of alertness, basically. And indeed, if we look at melatonin, so melatonin production starts here. So we have a peak, and melatonin production stops after that. So it's actually the exact opposite of what happens in core temperature. So if you plot these two together, you can see that they circle in antiphase. When melatonin is high, core body temperature is low, and alertness starts to decrease. And we've done this experiment many, many times, where actually we've given participants exogenous melatonin, because as you may know, melatonin is available in any drugstore now, at least here in the US. So you can ingest melatonin, and it actually reduces your feelings of alertness. So this is an image where we've given the same participant on different occasions, either a placebo pill, so they didn't know what was in the pill, or we gave them a melatonin pill, and it significantly reduced their alertness. So they felt much sleepier after ingesting melatonin. So there's a strong relationship between melatonin production and alertness. And I know Dr. Marijke Gordijn gave an excellent lecture about melatonin and alertness. So I would definitely encourage you guys to check it out if you're interested in this relationship. So the other thing that happens when you give participants melatonin is a reduction in temperature. So here in orange, we see the placebo condition, and relative to the placebo condition, people who ingest melatonin, they become colder. So they become colder and they become more sleepy. And this means, as I explained to you in terms of heat dissipation and heat constriction, if core body temperature becomes colder, the eye button, so the skin temperature that we place proximal, so here on the clavicles, which is a measure of what goes on in the core, becomes also colder. And what we measure in the hands and the feet, so where heat dissipation occurs, they become warmer. And this means that we really we become colder. So the core body temperature decreases, so proximal skin temperature decreases, we're colder inside, and it means that distally skin temperature increases because there's lots of heat dissipation going on. So what we pick up is an increased heat in the distal regions and colder temperatures in the proximal regions. And this happens when we ingest melatonin. So what happens if we expose people to a lot of light after melatonin ingestion? And this is where it becomes really, really interesting, because if we expose people to lots of light, so this is the light blue condition, actually core temperature goes up a little bit. So whereas under dimly lit conditions, the core cools off, so we become colder, under bright light conditions, we actually warm up a little. And why this is so important is because if people use exogenous melatonin, it's usually before they go to bed, so they're exposed to tons of light. And the efficacy of melatonin is going to be lower under high intensity lighting because core temperature doesn't decrease as much. So you don't become as sleepy, for example. So high intensity lighting increases core temperature after melatonin ingestion. And this means, as we saw earlier, that indeed in the hands, they become colder because the core becomes warmer. So proximal, so the skin temperature on the clavicles, heat conservation, it becomes warmer. This is the light blue line, which means that in the hands and the feet, that is mostly heat dissipation, they become cooler after melatonin ingestion and bright light exposure. So also thermoregulation has a strong circadian component and a strong homeostatic component. So again, we can use our forced synchrony design to really take apart the effects of the homeostat and of the circadian clock and how light interferes with these concepts. So if we look at the circadian clock, and here we have a bunch of parameters, including core body temperature, temperature measured at the forehead, so an eye button here at the forehead, a little sensor at the forehead, the clavicles, the navel, and we measured skin temperature at the hands, feet, toes, and the distal proximal gradient, which basically is a combination of these measures. And basically proximal, so what happens in the core body temperature cycles beautifully, so low core temperature after melatonin production, and higher core temperature during the circadian day. And we see a similar pattern in the forehead and a similar pattern in the clavicles and the navel, meaning that indeed, these are parameters of what happens in the core. If we look at the distal region, so where heat dissipation occurs, the hands, the feet, the toes, and the distal proximal gradient, it cycles in antiphase, meaning that when core temperature is low, the distal regions are high. So they are warm when the core is cool. So the hands, feet, and the toes, they get warm when the core starts to cool off. And we see some light effects here, but it's not dramatic, right? So sometimes there's a little bit of an effect of light in core body temperature here. There are some effects of light on navel temperature and especially in the toes. So it seems to be a little bit warmer, but nothing dramatic. So what if we look at the effects of homeostatic sleep pressure, so time awake? And here the effects become a little bit more dramatic. So while there's not really an effect on core body temperature, if we look at what happens in the clavicles, over the whole range, there's an increase in temperature, suggesting that people are warmer when they're exposed to high intensity lighting. And this also is shown in navel temperature. So it seems to suggest that there's heat conservation going on. And indeed, if we look at the distal regions where heat dissipation occurs, we can see that these are cooler. So the toes are cooler and the distal proximal gradient is cooler. So it seems to suggest that there's decreased heat dissipation under high intensity lighting. And the one thing that seems to be a little bit deviating is actually the forehead, because the forehead is cooler under high intensity lighting compared to the dim light conditions. So the forehead kind of seems to behave as a distal marker as opposed to a proximal marker. So taken together, what does this really mean? So the proximal skin increases under high intensity lighting, the distal skin decreases under high intensity lighting, but the core body temperature doesn't really do anything. And this seemed to suggest that there is some form of heat change going on, right? Like there's heat conservation going on, or heat dissipation going on, but nothing changes in core body temperature. And what this can indicate is that actually there might be a change in metabolism, because core body temperature is basically in part also a reflection of what of metabolism. And when you're metabolically very active, core temperature usually goes up. And here what we see is no change in core temperature, but a change in, but a warmer skin temperature in proximal regions and less heat dissipation in distal regions. And it might suggest that there's actually a change in metabolism. And for those who are aware, there's actually a high correlation by people conducting shift work and obesity. So it's possible that being exposed to high intensity lighting during your night shift is great for sleep, what I showed earlier, but it might not be so great for your actual metabolism, because it might be a sign that it decreases metabolism, and therefore you have reduced processing of food, which increases your chances of obesity. So for my scientific conclusions, the first part, light can influence subjective alertness at limited times of day, but light has profound effects on cognitive performance, which means that high intensity indoor lighting during the regular day might be beneficial for mental performance, even though it may not be perceived as such. So you may not know that you're actually performing better, even though you are. In terms of sleep, daytime light exposure improves objective markers of sleep. Daytime light exposure also improves self-reported sleep quality, and especially when the circadian clock is promoting sleep. Light can consolidate sleep potentially through increasing circadian amplitude. And this is something that I actually did not show this lecture. I apologize, this should not be here. And the last part of the talk about thermoregulation, exogenous melatonin decreases core temperature, and this may facilitate sleep onset. Bright light exposure induces vasoconstriction, so it results in heat conservation. And the lack of effect on core body temperature may suggest a decrease in metabolism, so less heat production. So this was a little bit about the scientific part of the lecture. And now I wanted to talk a little bit about my academic journey. And it's this beautiful quote: "We live in a world where the yellow brick road has many forks and can take us on many incredible journeys." And I think it's no different for at least my scientific journey, and I think for lots of people's scientific journeys. So maybe this will help a little bit, maybe for the early career researchers, to talk a little bit about how my road in academia looked like. Because when I started my PhD in March 2015, my topic of interest was really light effects on daytime human alertness. And the end product really, really significantly differed from this thesis topic. So my first experiment indeed was about the daytime effects on human alertness. And this is something that I talked about greatly in this talk. And I had non-significant results, right? Light did not induce any alertness during the day. And this made publication extremely hard. Non-significant results are notoriously hard to publish. And it actually took me 3 years to publish my first paper after starting my PhD, which I think is quite a substantial amount of time. And maybe more importantly, I was really at a crossroad, because my first experiment, which was really going to be a proof of concept, like, hey, light can induce alertness during the day, actually posed a really, really big crossroad in which I had to choose, like, okay, am I going to investigate alternative ways of improving daytime alertness? Do I want to investigate effects of caffeine, sleep quality at night, exercise, temperature, all these things that can induce alertness? Or should I try and figure out, like, why can light induce alertness at night but not during the day? So what is the underlying cause? So basically choosing between a more implicational PhD versus a more basic PhD. And in the end, obviously, after also consulting with my mentors, I chose to look at what are the underlying causes. And it led me through this whirlwind of experiments, because obviously when I present it like this, it seems like straightforward conclusions and logical experiments that follow each other, but it wasn't like that at all for me. My second experiment was about the effects of melatonin on human alertness, thermoregulation, and cognition, a little bit to find out like, hey, how do melatonin and alertness coincide? My third experiment was the big human force synchrony, in which we looked at the light effects on alertness, sleep, and thermoregulation. So the topics kept expanding. And the fourth experiment that I did not get a chance to talk about today is about the time of day effects on physical performance. So if we go back, and how my original thesis topic was about the light effects on daytime human alertness, in the end, I looked at alertness, yes, but also at melatonin, thermoregulation, cognition, sleep, physical performance, and it ended in a thesis being really about the mental and physiological responses to illumination. So it's definitely not the same as my initial goal. It ended up being something different, but I think it ended up being something so much better, actually, than I initially planned on doing. And of course, you know, the scientific journey comes with its amount of hardships, I would say. Like, yes, I think after my third experiment, I had my first real paper rejection, which is hard to deal with as a PhD student. And you know, the paper rejections don't stop. And I tried to propose for a couple of grants, and I had a grant rejection at the end of my PhD trajectory. And this was a very significant one, because it actually meant that I could no longer stay at the University of Groningen. I wanted to do a postdoc there, and there was no money, and my grant got rejected. So what was I supposed to do at the end of my PhD with no funding and no opportunity to stay at the University of Groningen? So it was really trying to decide, what do I want? And it turned out I mostly knew what I didn't want. I actually didn't want to live abroad, and especially not in the United States, because I had a lot of concerns about work-life balance. I like academia and I was passionate about science, but I knew I cannot stay at the University of Groningen. So these are, you know, opposites. I have to go abroad, even though I didn't really want to. And I knew I didn't have a future in academia if I stayed in the Netherlands, because if you want to become a professor, you need experience abroad. So these things were hard to reconcile. And in the end, I went to a conference, and I gave a presentation there, and the last slide of my presentation indicated that I was looking for a postdoc, because I felt like this is what I should do. I should at least advertise that I wanted to do a postdoc. And I met my current supervisor there, and the last slide of his presentation indicated that he was looking for a postdoc. So it was kind of, you know, a match made in heaven in a way. And I ended up moving to Stanford, even though I had my reservations about moving to the United States. I did it anyway, and I can say, like, I was very happy with my choice. And of course, you know, nobody can deny that I arrived in January 2020, so almost 4 years ago, and I arrived merely 6 weeks before the start of COVID-19 induced lab closures and the pandemic. So, I mean, it's been a tough road, I would say, and this will never stop, right? Like, this is just what the academic journey throws at you. And I continue to get papers rejected and grants rejected, but this is kind of the way academia works. So if I would say maybe the conclusions from my scientific journey, and you can take away from it what you like or what you don't want to take away from it, I think you can definitely stray from your original thesis topic, which might actually improve your thesis. I know it did for me. I think it's really, really important to stay open-minded. Like, if your results are not expected, yeah, what are you gonna do, you know? Adaptability and versatility, I think, are really key characteristics of a scientist. And I would say it's imperative to build a great relationship with your supervisors and mentors, and you have to rely on them. Like, I know I relied heavily on them after I got my non-significant results in my first experiment, and I had to decide what to do. I would really suggest visit conferences, talk to colleagues in the field, and be bold. I was very hesitant about putting this slide at the end of my presentation saying I was looking for a postdoc because I wasn't so sure that I actually was. And I did it, and it gave me great opportunities. And in the era in the realm of being bold, I'm also now going to put out there that I'm now looking for, the next step. I'm looking for professor position. So if you know anything, please let me know. And in the end, it's really about enjoying the ride. I think a life in academia is very up and down, but in the end, we have to enjoy it because it's also beautiful and cool and inspiring. So thank you so much for listening. And of course, all the work, it's not all me. I don't want to take credit for it. I really want to thank my current supervisor at Stanford University, Jamie Zeitzer, at the University of Groningen, my past supervisors, Domien Beersma and Roelof Hut, who I think is also part of the BioClock Consortium, and Tom Woelders, and from Chrono at Work, Marijke Gordijn. Thank you so much for listening.
Thank you very much, Renske, for your talk. It was really interesting, and especially for me as well, also the last part was definitely a good addition. Thank you.